Okay, I think we'll get started. Make sure our speaker has the allotted time. Before I get to introduce today's speaker, I just want to take a minute to acknowledge, I think many of you know that this week Dr. Ed Murphy died. Dr. Murphy was one of the two founders of this entire enterprise, former CEO of Carilion Health System, who worked very closely with Dr. Charles Steger, president of Virginia Tech, to found the VTC partnership. And so we all are going to miss Ed Murphy. I just want to say one thing I remember in many conversations with him. He said to me, hope is not a strategy, Mike, so come up with a strategic approach to things. I said something about hoping we accomplish something, so I always fondly remember Ed and what he did for this enterprise. So without further ado, I want to introduce this evening's speaker, Dr. Mark Baer from MIT. I've had the pleasure of knowing Mark and following his work for many years. I think we're maybe close to the same age. He's probably a little bit younger than me, I guess. But nonetheless, Mark is very well known for his work, and his work has followed a consistent thread, even though taking on different problems. But if you look at the entire body of work, you see the thread where he's really built from one area to another to make major inroads in neuroscience. Mark is the Picower Professor of Neuroscience at MIT in the Department of Brain and Cognitive Sciences. He held a Howard Hughes Medical Institute investigatorship for over 20 years, and that's got to be a record or close to a record. Most of the people I know did 1 5-year cycle or maybe 2, so that's obviously impressive and well-deserved. He's a fellow of the AAAS and also the American Academy of Arts and Sciences. He's received numerous awards for his work, including the Ipsen Foundation Neuroscience Prize, the Fragile X Foundation Research Foundation Pioneer Award, and also the American Society for Pharmacology and Experimental Therapeutics Fuller Award. Mark did his doctoral work at Brown University and got the highest acclaim. He was brought back after his postdoctoral training to be a faculty member at Brown and rose through the ranks there to become a full professor. After finishing his Ph.D., he went to work with Wolf Singer at the Max Planck in fundamental work on applying theoretical constructs, understanding experimental synaptic plasticity that has proved very worthwhile to many of us over the years. In 2003, he moved to MIT. He serves on multiple scientific boards, the Hopkins Lieber Brain Institute Board, the Scientific Advisory Board of the Fragile X Research Foundation, and has been a reviewing editor for Science magazine. Mark's earliest work involved studying synaptic plasticity in the visual cortex after visual deprivation. And from the very beginning, he was interested in modulation, various chemical neuromodulators and how they affected that plasticity, and published some very important work there, and then went on to work, as I said, with Wolf Singer, and carry that work on to another dimension, looking at all kinds of neurotransmitters, acetylcholine, norepinephrine, and also implicating a certain type of receptor that many of the students are familiar with, NMDA receptors, in plasticity of the visual cortex, and did foundational work there. But for me, with all the body of work up to that time, the stuff about Mark's work that I really enjoy is he took his very fundamental findings in the lab with mouse or rat and isolated preparations, and always insisted, was probably his own harshest critic, if you heard Mark talk at meetings, that we've got to show this in vivo. We have to understand what's happening in the living brain. how good is a construct when you've studied something in a dish or in the lab in an animal, and wanted to take it and do the hard experiments, challenging his own work and really developing hypotheses that could be falsified. And he did indeed demonstrate with his colleagues the importance of various forms of synaptic plasticity, such as what's called long-term potentiation and depression, as being necessary and involved with plasticity in the brain, in the visual system, and also in learning in the hippocampus. And then most recently, his work is really built on this theory that his lab developed and espoused called the mGlu-R theory of Fragile X syndrome. And he's not only gone on and taken the fundamental work on plasticity from the visual system and the hippocampus and moved it in to understanding this disease in animal models, but has moved on to attempt human clinical therapeutic trials. And they've had some really exciting findings over the last couple years. They've gotten close on some things. I think they're even closer right now. But it's a great example of how science should and can inform medicine and really make a difference in people's lives. So Mark really works across that intersection. I think he's a great example of the type of person we love to have come and speak in this series. So without further ado, please join me in welcoming Dr. Mark Baer.
Well, that was a lovely introduction, Mike. It's a pleasure to be here. In part, it's nice to see Mike again. We've known each other, I think it's 35 years or there, plus or minus. Hard to believe. Can you hear me okay? Yeah. So when Mike made the call, I had to answer it. And it's great to come down and see what you've helped to build here. Looks fantastic. I'm sorry I got here so late today, but so I should also mention I was born in Virginia, so it's always nice to come back to my home state. So this is the topic of the talk today, which is something about brain development. And brain development is a wondrous thing. And needless to say, a lot has to go right to build a brain. And we're by no means fully formed at the time of birth. And a lot of brain development continues right through adolescence and even into the ripe old age that Mike and I are at. We're still developing, right, Mike? Right. Unfortunately, it's also, because so much has to go right, it's also subject to disruption. And we would call those neurodevelopmental brain disorders. And we can sort of conceptualize this by imagining that there are many different functional domains of brain maturation. For example, vision or language or executive reasoning and so on. And these things will develop with different time courses. But when things are going well, there'll be some trajectory of acquisition of these skills due to the maturation of these circuits. Unfortunately, in disease states, this development gets derailed. And what do I mean by disease states? Well, what I mean is perhaps a consequence of a mutation, a gene mutation, perhaps it was an inherited or a de novo mutation. Another example would be diseases, for example, maternal infections. Another example would be deprivation, for example, poor nutrition or environmental deprivation when you're in infancy. In all these cases, development has been derailed. And the question we'd like to address is, at the time of diagnosis, which almost invariably is after birth and sometimes well after birth, can we devise treatments that can either prevent the further decline or actually reverse the damage that's been done? So that's really our objective here, is that, at this legacy stage of our careers, we'd like to be able to apply the knowledge that we've gained over the last 35 or so years and develop treatments for developmental brain disorders. So the disease I want to focus on today is called Fragile X, or Fragile X syndrome. And this is what you might call a simple neuropsychiatric disease, or a simple developmental brain disorder. What do I mean by simple? I mean, it's simple because we actually know the cause. We don't have to guess about it. The cause is silencing of a single gene in the genome called FMR1 that encodes a protein called FMRP. And all that goes wrong in the individuals with Fragile X is due to the absence of this FMRP protein. So it's what we would call a single gene disorder. So what goes wrong? Well, what goes wrong is manifest in many different ways. The sort of core manifestation is intellectual disability. So everyone that's affected by Fragile X is intellectually disabled, sometimes severely so. But in addition to intellectual disability, there are constellations of other symptoms, and some of them we might call autism-like. For example, social phobia, repetitive and stereotype behaviors, a poor eye contact. And other phenotypes would be something we might call hyperexcitability, like ADHD, increased incidence of childhood epilepsy, and so on. So although the cause is simple, the presentation is quite complex. So, of course, we now live in this era of molecular medicine. The human genome has been sequenced, and we can mine the genome for the causes of diseases, the genetic causes of diseases. And so there's a sort of roadmap that we hope to be able to apply to neurodevelopmental disorders. It goes something like this. We start with a well-phenotyped patient population that suffers from a psychiatric disease. And then by studying DNA from these patients, we identify the genes that are responsible. We can recreate those genetic lesions or mishaps or mutations in animal models. Then we can study those animals, and we can literally apply, the knowledge that we've gained over many decades of basic neurobiological research to understand how does the brain function differently in the face of these mutations. And this reveals what we would call disease pathophysiology. So not normal physiology, but the pathophysiology, how things are not functioning properly. And with a lot of luck, you may have an insight that suggests a potential treatment, some way to correct that pathophysiology. Maybe not all of it, but some of it, enough to make a difference in patients. So that's what we mean by target identification. And then if you find a good target, and particularly in the context of drug development, a target that you can use a drug to interact with it and set things right, then we'll go into hopefully introduce novel therapeutics after clinical validation and human clinical trials, and perhaps come up with a treatment for the disease. So that's what we're trying to do. There's also sort of a shortcut pathway here I should mention, which is very exciting, which is that you can actually take cells from affected individuals, and these can be differentiated into neurons in vitro. So you can actually study neurons that are actually derived from the affected individuals. And it's another route to get there, but you still have to understand disease pathophysiology. So where do we stand in Fragile X? Well, it's sort of a textbook example of the progression. And the story begins back in the 40s with the recognition by two clinicians, Martin and Bell, of a cohort of patients that seemed to present in a similar way, and they had a suspected genetic etiology. So originally, Fragile X was called Martin-Bell syndrome. And then many years later, when we finally developed the tools, thanks to the Human Genome Project, to actually understand what the genetic basis of the disease is, the affected gene was discovered by Steve Warren, Ben Ostra, and David Nelson. So that was in 1991. Shortly after the discovery of the gene, the animal model of the disease was created. So knocking that gene out, silencing that gene in mice, and more recently it's been done in rats and also in other more exotic species like fruit flies and zebrafish. So we have a number of animal models that we can then use to try to understand what's the pathophysiology. So in 2002, we made a discovery in my lab that was really an accident. We did not wake up one morning and say, hey, let's work on Fragile X. It was sort of, we were following our nose, and our path intersected with the Fragile X field. And we made a discovery that suggested the possibility that we might be able to correct aspects of Fragile X with a small molecule therapy. So this approach was validated, well validated in animals, and clinical trials were initiated. So what I'd like to do is to, let me begin with the best news. This is the good news. The good news is that we actually were able to correct Fragile X in animals with a drug therapy. I don't mean all of Fragile X, but a lot of Fragile X. Many symptoms of Fragile X in the mouse model could be corrected with a small molecule therapy. And this is, I'd say, the sort of milestone here. The breakthrough discovery or study was published in 2007, so that's 10 years ago. A lot of times these studies get published, and then there's usually a press release, and the press will pick it up. And the question is always, how many years till this discovery leads to a new treatment for affected humans? And the answer is always about 10 years. About 10 years. So okay, it's been about 10 years. So let's see where we are. I wish it happened faster. We tried, but it hasn't. But so this was really a life-changing experience for me. So what I wanted to do is I want to tell you about a little bit of neurobiology, how we got into the Fragile X field, what our discovery was, and then I want to tell you about the efforts that have been made to translate into human therapeutics. So there's a chance, I know this is a public lecture, there's a really mixed audience here. I may lose some of you along the way, I hope not, I'll try not to. But I want to make sure you get the take-home message right up front. So first, basic science can lead to breakthrough discoveries. What I mean is, seriously, we had no clue that our work would ever be relevant to a neurodevelopmental disease like Fragile X. We were literally following our nose, and it was just a chance discovery that led us into this field. It either, I don't know, you could say it derailed my career or defined my career. I'm not sure. You be the judge. Secondly, we live in this era of molecular medicine in which genetic causes of disease can be discovered, and these can be recreated in animal models that we can use to study pathophysiology. In the case of Fragile X, animal models have shown that significant benefits can be obtained even when you start a therapy after birth. That's a really important point, which is we think that it's in principle, it should be possible at the time of diagnosis, at say age 2 or 3, to actually administer a treatment that can alter the trajectory of the disease. However, translating discoveries in animals into humans is hard, expensive, and it occurs by trial and error. So do not expect it to succeed on the first try, and hopefully the resources and patience and interest is long enough or deep enough to actually ultimately succeed. In our case, we did not hit a home run in our first try, but we may have hit a base hit, a blooper in the infield, but nevertheless, we're still excited about it. You be the judge, I'll show you the data. And final point I want to make is that what we've learned in Fragile X we think may apply more broadly to other causes of intellectual disability and autism that have genetic roots. Okay, so here we go. Ready? Let's begin with some neurobiology. So our billions of neurons in our brains communicate at specialized junctions called synapses. So there's trillions of synapses in the brain. And at a synapse, nerve impulse enters the nerve terminal. Neurotransmitter is released in response to the arrival of that nerve impulse. Neurotransmitter binds to neurotransmitter receptors on the postsynaptic side, the other side of the synapse, and that can generate either biochemical or electrical signals, and this is how our neurons communicate with one another. There's a tremendous interest in synaptic transmission because we rely on it to function. If you block synaptic transmission in the brain, all brain function ceases, and we know that modification of synaptic transmission is the physical substrate of learning and memory. It's critical for the refinement of circuits during early development, and it's a process that goes awry in a number of disease states, including addiction and neurodegenerative diseases. So it's obviously attracted a lot of interest in the community of neurobiologists. So some of the neurons in the brain, I should say synapses typically, more often than not, are formed on dendrites. So these are these processes that are emitted by neurons. They act like the antennae that receive synaptic input from other neurons. And in many instances, these dendrites are decorated with these little structures that hang off of them that resemble the early anatomies of spines, so they're called dendritic spines. So this happens to be an image of living neurons that was made in my lab. These are two neurons that have been made fluorescent by expression of a fluorescent molecule. And we can sort of magnify this area of the dendritic tree, and you can see here these dendrites and all these little nubs that come off of it are dendritic spines, and dendritic spines are really specialized to receive synaptic input from other neurons. And if we take an even closer look at the dendritic spine, which would require an electron microscope, we get an image that looks something like this. So I've outlined, this is now shaving off a bit of that dendrite and look, a very thin section, and looking at the profile in the electron microscope. So the yellow line denotes the limiting membrane of the dendrite. This is the dendrite proper. This big dark stained thing here is a mitochondrion. And here's the dendritic spine that comes off, and I've shaded in blue the presynaptic side, that is the nerve terminal of another neuron that's communicating. Now, one of the interesting things about this particular image is this collection here of structures called ribosomes. Ribosomes are the site of protein synthesis. It's where messenger RNA is translated into protein. So this was quite a startling discovery when it was made quite a few years ago, actually made by Ozzie Stewart when he was at UVA. So this suggested that synaptic transmission, in addition to signaling electrically, can actually stimulate protein synthesis and whatever consequences there are of protein synthesis at synapses. For example, assembling new synapses. So this is sort of the cartoon view of what happens at a ribosome. So we have this collection of ribosomes. Probably these guys are collected together because many of them are bound to the same strand of messenger RNA. The ribosomes are assembled, and they scan down the messenger RNA, which is containing the information from the genes in the genome. So the messenger RNA goes from the nucleus of the cell out into the dendrite. It binds to these ribosomes. They scan along it, and in response, they synthesized a new protein molecule, so they can build new proteins. So what do we know about protein synthesis regulation at synapses? Well, it's quite a vast topic, actually. But some mechanisms appear to be more important than others. And just to give you a brief primer, what everybody in the world should know about synaptic transmission in their brains. The first thing you should know is that 80% of the synapses in your brain use glutamate as a neurotransmitter. Glutamate, simple amino acid, it's in the diet. It's used by most excitatory synapses in the brain, so about 80%. So there are many other neurotransmitters, but they're only comprise the other 20%. So if you had to pick one thing to study, hey, you should study glutamate. So glutamate is released in response to the arrival of a nerve impulse in the nerve terminal. So it spills out of these vesicles, and then it binds to postsynaptic receptors on the other side. And many of these receptors are what we call glutamate-gated ion channels. So when the glutamate molecule binds, a channel opens and allows electrically charged atoms to flow into the cell. And that's what gives rise to the sort of excitation part. So as electrically charged atoms flow in, it stimulates the postsynaptic cell, and that can eventually generate its own nerve impulses. So there's a number of different glutamate-gated ion channels, and I just want to call your attention to one of them that's called an AMPA receptor. It's a funny name, but it has to do with the chemical that binds selectively to that receptor. But we all call them AMPA receptors because we can't remember what AMPA stands for without looking it up. Okay, so glutamate comes out, it binds to AMPA receptors, channel opens, and you get an electrical signal. So that's the workhorse of synaptic transmission. If you block AMPA receptors in the brain, lights out, okay? Brain stops functioning. Even though those other 20% of neurotransmitters are there, you really can't tolerate the blockade of AMPA receptors in the brain. Well, in addition to these glutamate-gated ion channels, there are receptors that actually are not ion channels at all. They're what we call metabotropic receptors. And when glutamate binds to the metabotropic receptor, it actually generates a biochemical signal inside the cell. So it's signaling, but it occurs at a much slower rate. It's very local to where that receptor is. So what we've learned over the years is that there's one metabotropic receptor called the mGlu-R5 or mGlu5 receptor that when it binds glutamate, it activates a signaling pathway that leads to new protein synthesis at the synapse. This is a major mechanism by which activity in your brain is regulating protein synthesis at your synapses. There's a good chance it's very important for learning and memory. In fact, if you remember this lecture today, it's because your synapses are being modified, and in part, probably by synthesis of new proteins. So now I have to tell you a little bit more about how I stumbled into this area. So as Mike mentioned in the introduction, we have this long-standing interest in synaptic transmission and how it's modified by activity, okay, because of the big question of how do synapses modify to store information and to refine circuits during development. And the way that, one way to study this is to take a slice of the brain from a mouse or a rat, you keep it alive in vitro, and you can stimulate those synapses with a stimulating electrode. So you can put an electrode in here, and you can just give a little shock to evoke nerve impulses, and then you can record the responses with a recording electrode. And boy, I tell you, there are hundreds of labs doing this experiment right now, probably. Okay, they go in there, they stimulate those these nerve fibers every 30 seconds or so, and they record the response. And you can plot that data on a graph where you have the magnitude of this AMPA receptor-mediated transmission against time in the experiment. And in a good preparation, you get a nice stable baseline showing synaptic transmissions happy and stable. And as Mike mentioned, one of the things that we have been focused on quite a bit over the years has been a modification of synapses that makes them weaker. And we call that phenomenon long-term synaptic depression. So starting back in the early 90s, we've been studying long-term synaptic depression, and it's an interesting topic. And there are many ways to elicit it, but one way to elicit long-term synaptic depression is to activate the metabotropic glutamate receptor. So we can activate this receptor in a number of different ways. And what happens to this AMPA-mediated transmission? Well, it undergoes depression, and then that is quite steep decline initially, and then that will settle into some stable value, and we call that long-term depression. It's about 20% reduced in strength. But what Kim Huber discovered when she was a postdoc in my lab 17 years ago is that this response to activating mGlu-R5 is blocked by a protein synthesis inhibitor. So we were excited about this because we knew the machinery for protein synthesis was at synapses. There was some indication that activating mGlu-R5 could stimulate protein synthesis, and what Kim had discovered is one functional consequence of that, in this case, synaptic depression, which was surprising. We didn't expect to see synaptic depression requiring new protein synthesis, but it does. So the sort of cartoon then goes like this, that we release glutamate at these synapses, we activate the metabotropic receptors, this stimulates protein synthesis, and it triggers actually the internalization of these AMPA receptors, and so you get reduced synaptic transmission. So it's an interesting problem. Neurobiologists get excited about this stuff. So this, of course, then introduced a whole set of questions that we were ill-equipped to answer, but we were forced to, like how do mGlu-R5 receptors signal intracellularly? How is protein synthesis regulated at synapses? And what are these proteins that are synthesized? So it was really a universe of infinite possibilities of what experiment to do next. But the experiment that we did was to look at the potential involvement of the FMRP protein. Now, if you've forgotten about FMRP, I'm sorry, but that's the protein that's missing in Fragile X. Enter Fragile X. Why did we study FMRP? Well, there are a number of reasons why it was of interest, but I'll tell you the real reason. The real reason is I gave a talk describing Kim's results at a meeting, actually a Hughes meeting. I sat down after the talk, and the guy sitting next to me, who I didn't know, leaned over and congratulated me on the talk. I had mentioned Fragile X in my talk, I think, and he said, "You want to look at the Fragile X mouse?" And I said, "Sure, that sounds like a great idea." That guy turned out to be Steve Warren, who was the geneticist that discovered the mutation. So Steve generously sent us the mice, and so we did the obvious experiment. We looked at LTD. That's what we do. And this was the experiment that derailed my career. We discovered that, really contrary to our hypothesis, that the LTD response was exaggerated in Fragile X compared to the wild-type controls. We were actually expecting the exact opposite. So we had to think hard about what this meant. But what it suggested to us is the possibility that there's actually excessive mGluR-stimulated protein synthesis in the absence of FMRP. So we and other labs have addressed this question directly. It's actually fairly straightforward to measure protein synthesis. And the way you do that is you use radioactive amino acids, which are the precursors for new proteins, and you just feed your brain radioactive amino acids. You look at the rate of incorporation into protein, and then you can measure the radioactive protein molecules. And this was actually originally done by Carolyn Smith at NIMH, and she gave mice C14 labeled leucine in living mice, behaving mice, and looked at the incorporation of that leucine into protein. And what these are what we call autoradiograms, where the hot colors reflect increased radioactivity or increased regions of protein synthesis. This happens to be one of our favorite parts of the brain, the hippocampus, which looks kind of like the cartoon. So this is the labeling that occurs in the wild-type animals, and here's the labeling in the knockout. And you can see that there was an increase in protein synthesis. So it turns out this is quite a robust finding, and we can actually take the hippocampus out, prepare slices as if we were going to do electrophysiology, but then just feed those slices the radioactive amino acid, and we see about a 20-25% increase in protein synthesis. So this is a very reliable observation. Well, we now know that this is a well-grounded observation, because this protein FMRP is actually an mRNA binding protein. So FMRP binds messenger RNAs, and when it's bound, it prevents translation from occurring. So this is the major function of FMRP. It's got some other functions, but the major one is that it regulates protein synthesis. So normally protein synthesis is held in check by the binding of FMRP to messenger RNA. It stalls those ribosomes so they can't translate. In the case of Fragile X, the brakes are missing and you get dysregulated protein synthesis. So there's quite a consensus that the core pathophysiology in Fragile X is dysregulated or excessive protein synthesis. So I told you that in the brain at excitatory synapses, protein synthesis is regulated by the metabotropic glutamate receptor. So it occurred to us that maybe at synapses there exists a molecular machine that ensures that the supply of synaptic proteins keeps up with demand. So in this case, demand is registered as excitation of the brain, the release of glutamate, and activation of the metabotropic receptor. So that's calling for more protein synthesis. But as with every machine, it's necessary that there be regulatory mechanisms. And one of these regulatory mechanisms is provided by FMRP. So the analogy is that the mGluR5 receptor is like the accelerator on the car, and the FMRP is like the brake. And you really can't do without both of those functioning properly. So we imagine that there's some sort of a function that relates the synaptic performance or neural performance against the level of synaptic protein synthesis. And this function has this inverted U shape. So there's an optimal level of synaptic protein synthesis to ensure optimal neural performance, and that is ensured by these regulatory mechanisms, FMRP and mGluR5. In the case of Fragile X, as I said, it's like cutting the brakes. So in a sense, you're getting too much of a good thing. So there's excess protein synthesis. That red ball slides over to the right shoulder here and actually impairs performance of the synapse. So you can see now the logic behind, hey, what if we inhibited signaling through mGluR5 or the signaling through mGluR5, and could we slide that red ball back up to the top and see some restoration of normal function in Fragile X? So that was this became known as the mGluR theory of Fragile X. Extremely simple idea. It really is based on two assumptions. One is that that in Fragile X, there are exaggerated consequences of mGlu-stimulated protein synthesis, and that we can correct neurological and psychiatric symptoms by inhibiting that. So, is there any truth to this? Well, one way to address this is to use that assay of protein synthesis again. So remember, this is one where we feed radioactive amino acids to a slice. We see this elevation in Fragile X, and then showing here is 3 different manipulations that all have in common reducing mGlu-5. Okay, these are pharmacological manipulations over here. So acutely applying a drug that inhibits the mGlu-5 receptor could rescue this excessive protein synthesis. Interestingly, it didn't have any effect on the wild type, but it could correct the excess that we see in Fragile X. So assuming that this is close to the core pathophysiology of the disease, we might expect to see some benefit by inhibiting the mGlu-5 receptor. And as I told you, we do. We do see a benefit. So we've showed in 2007 that reducing the mGlu-5 gene expression by half could correct a large number of Fragile X phenotypes. And later we showed that chronic administration of a drug, even administered in adolescence, could reverse Fragile X phenotypes. So this is very exciting, but you don't have to take my word for it, because actually the whole community, the whole Fragile X community, and the mGlu-R community sort of rose to the challenge of testing this idea, because the implications are very important. So this is, I don't expect you to be able to read this table. We recently published in a review article, but it reminds me to tell you that these are 25 different phenotypes in the Fragile X mice, 25 different aberrant symptoms in Fragile X mice, all of which are corrected by inhibiting mGlu-5. And I guess the point is that we've also seen this in other species. We've seen it in the rat and the fruit fly model and the zebrafish model. Multiple independent investigators are involved. Many different phenotypic assays, as I said, and many approaches to validate that target. So many different molecules that have in common inhibition of mGlu-5 or the signaling downstream of mGlu-5. So in a way, it doesn't get any better than this. This is fantastic. So we're really excited. Oh, so it's a great time to be a Fragile X mouse. And what's more, if that wasn't good enough, I mentioned that individuals with Fragile X, many of them have autistic features. In fact, it's often said that Fragile X is the leading known heritable cause of autism. It's not a major cause. Autism is a complicated disease, but this is the genetic risk architecture of autism, and you can see what the biggest, just about the biggest slice of pie here is Fragile X. And there are other gene mutations that are known to lead to autism and intellectual disability, such as that responsible for Rett syndrome, or another disorder called 16p11.2 microdeletion syndrome. And so when you find one thing that's wrong in a rare disease, you can sort of broaden your lens a little bit and ask, well, do other genetic lesions have in common this altered protein synthesis regulation? So there's some commonality, despite the fact there are hundreds of causes of autism. Maybe a substantial fraction of those have this same pathophysiology. And it appears that there's some merit to that argument. So we've done some studies in the mouse model, the 16p11.2 microdeletion, and in the mouse model of Rett syndrome, and have shown, again, that inhibiting the mGlu-5 receptor can confer benefit. So it's not necessarily as dramatic as what we saw in Fragile X, but it seems to be moving in the right direction. So, wow, this is great. Here we are. The disease pathophysiology is exaggerated consequences of mGlu-5 activation, and I should mention there are some really good drugs because the pharmaceutical industry invested very heavily in mGlu-5 as a target. They're not perfect drugs. They have warts, but they're pretty good. They invested a lot of money in developing these, and they sponsored clinical trials. So we just got the results of the clinical trials the first real round of placebo-controlled clinical trials, so-called phase 3 clinical trials, just published last year, 2016. What do you suppose happened? mGluR5 trials failed to demonstrate benefit. And of course, not only were the researchers anticipating success and the families anticipating success, the press, the medical press was anticipating success. So when these results were published, there was a lot of press articles published about this profound disappointment that we didn't succeed. So what's going on? And I guess the real question is this the death of the mGluR theory? Okay, it was great while it lasted, but hey, after 15 years, is it gone? I do not believe that. There are those of you, there's a sentiment, particularly in the pharmaceutical industry, that mice have sometimes not been predictive, oftentimes, not been predictive of human therapeutic efficacy. And it's for sure true. Mice and humans are quite different. Our brains are far more complex than a mouse brain. However, I don't buy this argument. For one thing, is we saw an evolutionary conservation of the relationship between FMRP and the mGluR5 receptor in mice, rats, fruit flies, and zebrafish. So I do not believe we're the exception, but I don't think we are asking the right questions. So another thing this is all new to me because I am a basic scientist, so I was sort of learning on the fly about how difficult it is to design and interpret clinical trials. So this is a literal back of the envelope calculation. I often like to point out that this is an envelope from the Internal Revenue Service that I put to good use. And I just want to just sort of sketched out some of the decisions that have to be made when you launch a clinical trial. And of course, none of us have a crystal ball, no matter how smart we are. So you have to assign a certain probability that you're going to guess right. So I'm a pretty confident guy. I gave myself an 80% chance that I would guess right. So 4 out of 5 shots I get the right. So here's some of the variables. Like, is mGluR5 actually the right target? Are we using the right molecule? Is the dose correct? Is the duration of dosing correct? Do we have the right patients? Are they the right age? Have we sampled from enough patients to see an effect? Are we using the right endpoints for therapeutic efficacy? And in the end of the day, when you multiply all these probabilities together, you find that actually your odds of success in any one clinical trial are pretty low. So it's very rare to succeed on the first try. It kind of reminded me of Warren Buffett's famous offer to pay $1 billion to the lucky basketball fan that could guess the outcome of all 32 NCAA basketball games during March Madness. And of course, it was the safest bet he'd ever made because nobody could possibly do that. Well, you could, but the odds are infinitesimally low. So there are issues in the clinical trials. And actually, in the case of the mGluR5 trials, right off the bat, one of the issues is the youngest subjects were 12 years of age, which is a long time to have developed with Fragile X, and then expect to see a substantial benefit with only 10 weeks of treatment. That's really asking a lot. And the reason that they started those trials so late in age was just because those compounds had never been in humans before, safety wasn't really known, and they really couldn't start in a pediatric population. There are other limitations, like you don't really have an objective measure of whether the drug is working, so-called biomarker, and the assessment tools are extremely crude. They're literally questionnaires given to the caregivers or to the clinicians. So we don't actually measure anything directly from the patient. It's more observations of the patient. And one of the things that we've learned is there are huge placebo effects because the parents are anticipating success. And so that actually yields a strong placebo response. The mGluR5 receptor itself, we now understand better, had some warts. All drugs have dose-limiting side effects. Everything, water, aspirin, has a dose-limiting side effect. And the same thing is true with the mGluR5 inhibitor. But that puts a limit on the level of exposure. So one would like to optimize that therapeutic window. And something we've recognized only recently is that there may have been quite a bit of tolerance to the drug. So you know how sometimes if you're taking a medication, it loses effectiveness over time. And this was probably observed in that trial, and we have animals data to support that. So, okay, we didn't get there the first trial, but we learned a lot. So these trials were the first placebo-controlled trials ever done in Fragile X, and Fragile X is leading the way for all other neurodevelopmental disorders. So we're really at the, as my colleague says, it's like building the plane while you're flying it. So we had to make a lot of guesses and hope hope for the best as you're moving along. But the best you can do is to learn from what you've done and then try to apply that knowledge to get it right the next time. So I wanted to tell you in the last few minutes about another drug development effort that is really targeting the same process in Fragile X, but by a different mechanism. So let me reintroduce the synapse to you. Here's a synapse. We know normally we stimulate mGluR5 receptors. We stimulate protein synthesis, and that's held in check by this FMRP protein. In Fragile X, the FMRP protein is missing. We get exaggerated consequences of activating this receptor, and that wreaks havoc on synapses and brain function. And I had argued that we could inhibit that by inhibiting mGluR5, but there are a lot of other approaches that we can take. And one of them is actually to go upstream and inhibit the release of glutamate. And a way to do that is with another drug called arbaclofen, which activates another receptor called a GABA B receptor. So GABA B receptors GABA is the major inhibitory neurotransmitter in the brain, so that's another 15% of your synapses. Use GABA. And arbaclofen is a activator of the GABA B receptor, and that is known to suppress glutamate release at excitatory synapses. So it's a couple steps removed, but the reason arbaclofen is of interest is that it's a compound that's already used in pediatric populations. So not arbaclofen, but baclofen. Arbaclofen is just one of the enantiomers of baclofen. But baclofen's been around for a long time. It's used to treat spasticity in cerebral palsy and also in multiple sclerosis. So there's a great deal known about baclofen and what its safety profile is. So you can actually go into younger subjects with baclofen or the active enantiomer. And we had shown in studies in the mice that baclofen can also restore normal protein synthesis. So it's another shot on goal. So I was actually involved in these trials because these were sponsored by a small drug company that I founded called Seaside Therapeutics. And actually, this was the very first ever placebo-controlled study done in Fragile X. And so this is what's called a phase 2 study. So it's an exploratory study. You don't really expect it to succeed. You hope it will, but you don't expect it. You expect to be able to learn from it. So the design of the study was the subjects come in. All subjects have Fragile X. And they're divided into two groups. One group is going to get placebo during the first arm of the trial, and the other group is going to get the active compound, which they called STX-209, which is arbaclofen. So then you follow those subjects for the first arm of the trial, then you wash it out, and then you reverse, so that every subject will see placebo and the active compound. So that's how this so-called crossover design. And the assessment instruments are, as I mentioned, very crude. And many of these decisions about how to assess really are based on regulatory precedent. That is, what measures has the FDA used to approve medications before? Not necessarily the most, sort of scientifically driven endpoint choices. And they're crude. So one of them is clinical global impression. A physician just makes an assessment of whether the subject is improved on the drug or placebo. Another is a so-called visual analog scales, where the parents will nominate 3 problem behaviors, and then on a sliding scale rate whether it was moved in one direction or another based on the treatment. And finally, something called the aberrant behavior checklist, which is a questionnaire that's given to the caregivers They fill out this questionnaire, and then there's a factor analysis done, and there are various subscales. And one of them, the most common one, is the irritability subscale. So it's called the ABCI, the irritability subscale. And another is social avoidance. So what happened? Well, okay, this is a table. It's not that visually appealing, but I wanted to point out a couple of things. So I want to draw your attention to this ABCI. And the first reason I want to draw your attention to this is ABCI was declared as the primary endpoint. So whenever you do a clinical trial, if you want to get FDA approval, you have to, a priori, before the trial begins, declare one measure as the one that's going to determine whether the drug works or not. That's the primary endpoint. If you hit that, you have a successful trial. If you don't hit it, you have a failed trial. So again, based on regulatory precedent, the aberrant behavior checklist irritability subscale was chosen as the primary endpoint. Now, when that number goes down, there's an improvement in irritability. So now I want to draw your attention to subjects treated with placebo. So here's the baseline score at 21.8, and the placebo, after placebo treatment, you can see it went down to 16.2. That is a placebo effect. That is anticipation of improvement by the caregivers. And there was really no difference on the drug compared to placebo. So this was a failed trial. However, there were some other measures that looked quite promising. So one was, for example, these visual analog scale of problem behaviors. There was a fairly dramatic improvement, and that reached statistical significance. And perhaps most exciting was this aberrant behavior checklist social avoidance subscale, which actually had been introduced only after the study had published, but the analysis was done again. That's why it's called a post hoc, because it wasn't proposed going into the trial, but we had the data and we could calculate it. And there we saw, again, a highly statistically significant improvement in this subscale. So very exciting, very good news. So on to phase 3. So phase 3 means usually it's a multi-center trial, and if it's successful, it will get FDA approval. And so this was recently published just a couple of months ago. So again, a busy slide, but makes the point that a lot of work was put into this. And actually, one thing I forgot to mention about this crossover, pardon for going backwards, is that because it was an exploratory trial, there was really a broad range of ages and drug doses, okay, because we were just exploring this space, but really an age range of 6 to 40, which is quite large. So in this phase 3 trial, the subjects were divided into age groups. I'm not going to tell you about the adolescent and adult study because it simply didn't work. I'm going to tell you about the study that was done in the 5 to 11-year-olds. And those subjects were divided into 4 groups, placebo, and 3 different fixed doses, escalating doses of the drug. And I can tell you the only dose that worked was the highest dose. So another thing we learned is, wow, maybe we haven't been dosing high enough. But I'll show you some of the data from the high dose group. So this is looking, and the primary endpoint in this trial was social avoidance because of the phase 2 trial. So here are the data for social avoidance in the high dose group. So again, improvement is when these numbers go down. The placebo is in red. You can see, again, this doggone placebo effect. And there's some differentiation apparent in the drug-treated group, but it didn't achieve statistical significance, so this is a failed trial. However, to our surprise, the irritability subscale did show quite a dramatic improvement. So here's the placebo effect again, and you can see that the arbaclofen-treated subjects really differentiated nicely. You could ask, why did irritability respond in this trial and not in the first trial? So of course I don't actually have the answer, but our suspicion is that in this trial, because we're talking about children, they're actually far more irritable. So it's really 2 things. They're children, so we expect children to respond differently anyway, because they're more modifiable. But secondly, they're more irritable. We know the scores at baseline were quite much higher, like twice as high as what we had seen used in the previous drug. So here's another way of looking at that. So this is the improvement on placebo and on the drug-treated group, again, statistically significant. And one of the reasons of course, you never know. If you measure enough things, you'll get a statistically significant effect that may be spurious. But one of the reasons we actually believe this result is there's another independent measure, not of the children, but of the parents. It's called the Parenting Stress Index. And you can see there's a very dramatic improvement in the Parenting Stress Index. So those involved in this trial really believe that this drug conferred benefit. So what do these numbers mean in a human way? Like, what does it mean that they're less irritable? And it's a really moving interview done with the mother of a young boy that was involved in these trials. And I'll just read what this says here, which is, a geneticist gave Holly the grim prognosis. Parker would live in an institution. He would never be toilet trained. He would never go to school. He would never be independent. Undaunted, Holly enrolled Parker, then 10, in one of Seaside's trials of arbaclofen for Fragile X. At the time, Parker could only say a few words. Instead of talking, he screamed. He had a happy scream and a sad scream and an I want that scream, Holly says. School was an exercise in frustration. If he had a rough day, he'd lunge at me and bite and not let go. She had huge welts and really bad bruises on her legs and arms. At first, nothing. Then about 2 weeks after Parker switched from placebo to the arbaclofen, he knocked a glass off the kitchen table. As Holly turned to pick up the shard, this always breaks me up. As Holly turned to pick up the shards, she heard her son say, "I'm sorry, Mom, I love you." And it's the first time he had uttered those words, and they came out in complete sentences. So that's what we're trying to accomplish. I can't believe I still get choked up about this, but I do. So just to return briefly again to the take-home messages, just to tell you where we've gone. Hopefully you're convinced that basic science is a good investment. We hopefully are living up to the promise of basic science and the promise of molecular medicine. The animal studies have shown, and it's very important, this is a genetic disorder. There are many things that could be messed up with brain development. But what the data suggests is that many of the symptoms are due to ongoing differences in signaling. And if we understand those differences, we can correct them even after birth. That's an extremely important insight. And we've shown now, again, in many different mouse models of different genetic diseases, that it's possible to intervene late. Translation is hard. It's very hard, and it's very expensive. And the real tragedy here is if we run out of money or commitment before we can actually track down some of these leads. We did not hit a home run. I would suggest that our arbaclofen data represent a base hit. We will see. And I think, again, what we've learned in Fragile X may apply to other causes of autism. So, on a happy note, I've told you about mGluR5 inhibitors and arbaclofen. There are many, many arrows in the quiver or shots on goal. Over the last 10 years, we've learned a tremendous amount about the signaling pathway that couples this receptor to protein synthesis. And there are, unfortunately, despite these, the fact that no one has yet succeeded, there's tremendous interest in the field. So there are a lot of clinical trials underway with different targets done by drug companies, mostly small. The occasional giant is still in the ring. So we're really very hopeful that we're going to get there. And, when I got these data on the arbaclofen and I was sort of looking at my shoelaces, I happened to be at a meeting at the Salk Institute. And I was just walking along, shuffling along, and there right on the sidewalk is this saying from Jonas Salk that hope lies in dreams and imagination and the courage of those who dare to make dreams into reality, and I would say to dare to persist to make those dreams into reality. And finally, I just want to thank, these are the people that really are responsible for the clinical work, and I've gotten generous funding from a large number of organizations. And finally, this is a slightly dated photo, but I'm not going to name them all, but just to make the point that I have done none of these experiments not one. These experiments are all done by really talented graduate students and postdocs. So that's it.
Great, Mark. Thank you very much. Inspiring talk. We have time for some questions. Carissa.
Is the specificity of RNA binding by FMRP known, and is it a few key proteins that are subject to that translational regulation, or more of a pool that are having an effect on the synapse?
Yeah, it's really a good question. It's a burning question, and a number of approaches have been taken to address that. But I think at the sort of at the most elementary level, we know that about 900 mRNAs bind FMRP. So it's not just a handful of FMRP targets. And it's even more complicated in the animal where, which of these transcripts are giving rise to that altered protein synthesis rate. Many of those aren't even FMRP targets. So there's secondary consequences of the loss of FMRP. So we haven't really narrowed down, focused in on the key pathogenic protein. The one that's attracted the most interest, however, is a protein called MMP9, matrix metalloprotease 9. Somebody probably works on that now. Right? You work on that? MMP9 is pretty, it's exciting. There are drugs that inhibit it, and there have been some success in Fragile X. Yeah.
I wonder if you could account for some of your clinical data by looking at, by bracketing off some of your patients according to the number of CGG repeats they would have.
Yeah.
And so instead of like working with a lot of dose variation, look at the level of pathology.
Yeah. These are all full mutation. So so all subjects have fully methylated promoter regions in FMR1. However, so we don't have that issue. However, individuals with Fragile X are typically mosaic because the gene isn't silenced immediately. It only silences after at about 12 weeks of age. So there are neurons that express FMRP. And what we don't know, and I'm very interested to have the answer to this question, is what regions of the brain are more affected, how much variation is there from one region to the next. I think if we understood that, it might explain, we might be able to map that better onto the symptoms, because the clinical presentation is quite variable. So.
Given that, sorry, let me make sure I'm pronouncing the drug right, arbaclofen, since you saw improvements in children as opposed to adults, have you gone back into the mouse model at all to look and see if starting administration of the drug at an earlier time point then allows for the adult mice to show improvements that maybe would not be there when it's given?
No, we haven't. It's a good idea, though. But we haven't done that. In the mice, we see good effects with these treatments quite late. So there are some interesting differences between those of us who study developmental plasticity. We realize mice have a lot of juvenile plasticity late in life, surprisingly. So that could be a species difference of when your window of opportunity is. But we've not actually done any systematic investigation of that. Yeah.
Do you see, do they regulate across the blood-brain barrier, these compounds? And the second question, have you seen an allometric scaling effect so that actually what looks good in the mouse doesn't translate as those species you get more species like humans?
Well, let me answer the first question that I know the answer to. So the mGlu-R compounds for sure do, because they're good PET ligands. So there's no uncertainty about whether they're in there and engaging the target, which often is the explanation for a failed trial. The same thing is true with arbaclofen, even though there is no PET ligand, so it's more empirical. The fact that arbaclofen's been used for many decades, or baclofen's been used for many decades, so it's clearly got CNS effects. It's a highly polar molecule. It doesn't have the same ready access to the brain as other compounds. As far as allometric scaling goes, I think it's an imperfect that's an imperfect science, I would argue, not being an expert. So, frankly, I don't know what went into the decision-making process about what were going to be the initial dosages used. I think that with respect to the arbaclofen, the side effect that we were alert to was sedation. That seemed to be the one to be most worried about. None of these kids showed any sedation. In fact, some of them were more alert, probably because they were sleeping better at night. But so that's that. It's just, as far as I know, it's just been done empirically. Yeah.
Since it is FDA approved, do you have any idea of the off-label use by pediatric neurologists for kids with Fragile X now?
Yes, a little bit. So when one of the sort of heart-wrenching things about the arbaclofen trial was that all the subjects in those trials had continued on the drug. They were given the opportunity to continue on the drug, and the vast majority stayed on the drug. So there were some Fragile X kids that were on the drug for well over a year, 2 years, and reporting great benefits by the parents. And so Seaside unfortunately ran out of money, and they had to discontinue providing the drug. And so it was a really horror story about these kids had to be weaned off of the arbaclofen. And of course, the pediatricians were substituting racemic at 2x the dose to see if that would have the effect. I hear different things from different people. It seems like it's clearly inferior, baclofen is to arbaclofen. There's something about the S enantiomer that probably is interfering with the active actions of racemic. But some have stayed on racemic and done reasonably well. So it's a mixed bag. Yeah.
We have time for one more question. Yep.
So I'm interested in your recent work that you published with Lawrence Stoppel looking at the ARV2 FMR1 knockout mice. And I was just curious, do you have any molecules in mind, compounds to interfere with the beta-arrestin signaling that might go towards clinical trials?
Yeah, wow, awesome question. Awesome. Thank you for that question. So just a little, the short answer is no, I don't have any compounds. But just to fill in a little background to your question. When we had the failure of the mGluR5 trials, we've been going back and saying, were there any red flags that we should have seen? Obviously, we were all very optimistic. And so one of the observations in humans is that at some doses that there'll be hallucinations. These are in normal volunteers. And so that obviously sets the upper limit on the drug exposure. And those can be very severe, in fact, depending on the molecule and the pharmacokinetics. So we didn't appreciate this fact, but if you go back in mouse, you can see that it will elicit behaviors at some doses that what we would call psychotomimetic in the mouse model. So we're like, oh, wow, that's there. So the question is what's responsible for those symptoms? And this is a deep question, but the metabotropic receptor actually signals by 3 distinct signaling pathways, and only one of those was used to screen for those compounds that were used clinically. And it turns out that's the pathway that's responsible for the side effect. There's another pathway using beta-arrestin. Its separate pathway is responsible for therapeutic effects. So one of the trends in pharmaceutical science or pharmacology right now is to develop what's called biased ligands that can actually selectively modulate one signaling pathway over another downstream of these receptors. So we don't have those molecules. I'm told that those molecules undoubtedly exist in libraries of pharma companies. And one of our hopes of publishing the paper was to attract their interest enough to do that. And my collaborator on that study, Bob Lefkowitz at Duke, who's a Nobel laureate in chemistry, I think is going to start a company to do this. So I'm glad about that. Yeah.
I noticed it was the last question, but I see a student with a minute. One more chance. Last question. Go ahead. Yep.
How much is
Cannabidiol, right?
Yes, sorry for the wrong.
Yeah.
That actually is really good to like resistant treatment seizures and also like anti-stress characteristics. So I was wondering if that could maybe help Fragile X syndrome, or if there is any model out there using it.
That is a good question, and there are people working on that. Actually, Kim Huber, who was my former postdoc and now has a fantastic lab in Dallas, has been doing some studies on cannabinoid signaling. So there is a connection. In fact, there's always been a suspicious connection between the metabotropic receptor and cannabinoid signaling. So yes, it's an active area of inquiry, so a lot of interest in that. We don't work on it in my lab, but.
Great, please join me in thanking Dr. Bear for
You're still awake. I'm impressed.